ECO-FCE project started in 2013, in January 2013, and will conclude at the end of 2016. The main goals of the ECO-FCE project were to improve the feed efficiency of pigs and broiler chickens and also reduce their ecological footprint. So working with 17 partners from across Europe, representing 9 different countries, we worked on this project and those main goals were delivered across 5 different work packages. So just to review the main findings from those work areas and how they will advance knowledge going forward and how they potentially could be used going forward. The first work package, or the first work area, was really to collate all the knowledge we currently know within the open literature with regards to feed efficiency of pigs and broiler chickens. And what we faced there, we collected a lot of information. We collected over 1 million references and tried to condense those down into a manageable resource. But what we found was there was a very large variety of quality and quantity in these open literature papers, and they didn't always tell us exactly what we needed to know to fulfill our database of information. As a result, we have got some pointers coming out of that. We have written a paper to look at novel feed ingredients which could be of use in the future for improved feed efficiency. But a big finding from that piece of work was the fact that the quality of information in the open literature doesn't lend itself very well to a large-scale meta-analysis and the utilisation of this information on a very large scale. So that has helped us develop our thoughts and what we'd like to do in the future, using the knowledge we've gained from the exercise itself, is to develop a more organised, sophisticated model for capturing this information from peer-reviewed journals with the foresight of them being able to use it more strategically in meta-analysis for companies and researchers going forward. Second very large work package focused on feed strategies for pigs and broiler chickens. And this work was largely driven by IRTA in Spain, with AFBI and Chugus being other partners in it, as well as the feed companies of ECO-FCE, Definition Nutrition, ITPSA in Spain and Delacon in Austria. So within that work package we had a large focus on enzymes, phytogenetic feed additives, the processing of cereals. A significant part of that work package was the focus on actual bench studies to try and predict the performance and efficacy of enzymes based on different raw materials. And that was conducted with IRTA. And we found from that piece of work that enzymes were very specific to the raw material of use. And we had very mixed results with regards to the efficacy of enzymes using these bench studies. But surprisingly, one of the key results coming from that was the effect of protease enzyme on peas and beans, which seemed to be very positive. So using that knowledge, we completed a piece of work in Chugus on actual live animals to test the efficacy in a real diet on real animals. And indeed, we did find improvements in feed efficiency and an effect of the protease enzyme in the pea bean based diet when it was fed to the animals. So that's a very positive result for the project, and indeed informs the enzyme industry going forward about the efficacy of the protease enzyme in particular on different substrates. In addition to that, we did trial a large range of other enzymes on various feed ingredients, and one of our main focuses was to look at rapeseed meal and distillers grains as suppliers of protein as opposed to soya. So across a few pig and poultry trials, we formulated diets with very low soya content and therefore a significant proportion of rapeseed and distillers grain content, and added to those diets varying levels of enzymes. We had quite disappointing results from that work in that enzymes didn't always work and there was very mixed results. So a key message from that is really straight down to the farmers and for farmers to make sure that the enzymes that are going into their diets do work. And the message there is to farmers to say enzymes need to be used in a very targeted approach against specific raw materials as opposed to a blanket approach. Another piece of work that we looked at very much focused on the gut and its role in how to improve feed efficiency in pigs and broilers. And within that gut work, we looked at both the gut structure, its immune status, and very significantly, its microbiota content. So a very unique piece of work that we conducted in that across pigs and chickens was an experiment which was conducted at Chuggas in Ireland, VetMed Uni in Austria, and AFBI here in Northern Ireland. We had the same protocol across all 3 sites, so therefore we were able to do the same experiment across all 3 sites with similar terminal sire genetics, similar feed formulations, but environment did differ across the 3 sites. From that experiment, we monitored the feed use efficiency of pigs and chickens, and we were able to determine animals with a very good potential for feed efficiency or a poor potential for feed efficiency. We then used those animals to dig down deeper into the common factors within the gut that was driving that high potential for feed efficiency or poor potential for feed efficiency. So what we found from that on the chicken side is actually location does have a very strong effect on the performance of the animals. And there were only a few biomarkers at the gut level that were common across both sites for high and low feed efficient birds. With regards to the pigs, a similar story. We found there was a very strong location effect, but as well as that, there were a few biomarkers at the microbiota level and the gut level which would predict high and low potential for feed efficiency. But on the pig work, it was also very apparent that the mother's microbiota had a very significant impact on the animal's microbiota throughout life. So using that knowledge, we started to look at the impact of the mother's microbiota on lifetime performance of the animals. And indeed, we found in a controlled study how strong that effect can be. In our study, we actually reduced the feed efficiency of the subsequent animals, but did prove the point that early life nutrition and early life exposure to microbiota from the mother is a very important determinant going forward. The genetic composition of the animals was then the third major area of work within the project. And from the genetic perspective, we also looked at the common denominators between high and low variable animals, but we also worked with COB as well as Hermitage genetics to try and develop SNP chips and specific genetic regions for coding for feed efficiency. As you can imagine, feed efficiency is the combination of feed intake as well as growth performance, so there's no one area of the genetic region coding specifically for feed efficiency. It's a combination of different areas. And our goal was to try and find what the best combination was. So using 1,000 animals generated from the COB facilities and 1,000 animals generated from the Hermitage facilities, we were successful in identifying some genetic codes. And indeed, very much a success story from ECO-FCE is the fact that for the Hermitage animals, they have now developed and are considering adopting more of a genomic approach using these SNP chips that were developed in their breeding programme going forward, and that should accelerate their rate of success in breeding for improved feed efficiency. So overall, ECO-FCE has learnt us the feelings of our current portfolio of, has learnt us about the current feelings of our current open literature and how that can be fully utilised, but it has learnt us how to then potentially utilise it better. It has taught us about the efficacy of enzymes and how they need to be very targeted, and also it has provided some very robust independent evidence to industry and particularly farmers on how to use enzymes properly. And it has very much advanced our knowledge of the common denominators from a gut microbiota, gut structure and genetic perspective of animals with high and low feed efficiency. And lastly, it developed genetic tools to improve the breeding for feed efficiency.